There's the very cute animals. Okay, back to microbiology. Can't wait for my stand to arrive. It's going to be so much easier. Okay, we're starting with humoral immunity, which relies on the activation of B cells. Make sure this is actually in view. There we go. Antibodies belong to a class of proteins called immunoglobulins, LG, in the shape of a Y. The FC, which is the tail of the Y region, determine the type of immunoglobulin, for example, IgM, IgD, etc. Alright. IgG is the most abundant in circulation. It promotes phagocytosis, cell lysis, confers passive immunity. IgA is present in saliva, tears, breast milk, most abundantly produced in general. IgM is secreted early, promotes agglutination, phagocytosis, and cell lysis. IgD is a surface antibody on B lymphocytes. Role is unclear. Not underlining that. IgE is important in parasitic infections and some allergic responses. Oof, I don't remember this FAB section, FAB something. The arms of the Y is responsible for antigen binding specificity. Antigen binding. That makes sense. Antibodies bind to antigens, thus promoting their destruction directly by precipitating soluble antigens, lysis of membranes, agglutination, which means clumping together, neutralization of toxins, or inactivation of some viruses. Let's take a look at this chart on immunoglobulins. IgG is a monomer. It's the smallest. Secondary response, most abundant. Yes, complement. Yes, crosses the placenta. IgA is a dimer. It's the largest. Big A. Found in secretions like saliva, tears, and breast milk. No complement. Does not cross placenta. So IgG is the only one that crosses the placenta in this chart that I see. So IgM, pentamer, largest, biggest M. Primary response, first in the fetus, no complement. So none of the others have complement or placenta crossing. And then IgD is a monomer. It's smaller. Activates basophils and mast cells. IgD, basophils, mast cells. IgE is monomer, smaller. Type 1 hypersensitivity parasitic disease. So parasites E. Try to eat you. Cell-mediated immunity. Relies on activated T lymphocytes, important in combating viral and fungal infections, as well as against potential cancer cells. T cells are activated by exposure to foreign antigens, which are bound by specific receptors. 3 types of T lymphocytes, which with their different functions. Helper Ts are the most numerous. They're activated by, fix the camera. Activated by macrophage processed antigens. Once stimulated, lymphokines, interleukin 2 through 6, and interferon are released. T lymphocytes, most numerous, activated by antigens. Oh, please don't bark on my video. I really don't want to start a new video. Sunny, lie down. You barker. Lie down. Okay. Cytotoxic killer T cells. They lyse the cells carrying the antigens to which they are sensitive. Lymphokine activated killer cells, try and talk over my dog growling, are activated by lymphokines. Suppressor T lymphocytes inhibit lymphocytic function. Cell-mediated responses persist longer than antibody responses. Natural versus artificial and active versus passive immunity. All right. Natural active immunity involves the production of antibodies after infection. Natural passive occurs when the antibodies are produced in the mother and transferred at birth to the infant. Artificial active immunity, the antibody production is induced by immunizations. That's what we're all hoping for now in the world. Artificial passive immunity, the antibodies are produced elsewhere, like in horses, and injected or transferred. Natural active and artificial active, longer lasting compared to natural passive and artificial passive. That last description is sort of lacking in my opinion. Hypersensitivity reactions. Can use the acronym ACID apparently, but those don't really help me, so I'm just going to take it one at a time. Type 1, anaphylactic. The antigen reacts with IgE, causing mast cells to degranulate. Release of histamine is seen in allergies like bee stings. Type 2, cytotoxic, IgG, IgM, destroys cells, erythroblastosis fetalis. Acute transfusion reaction, autoimmune, hemolytic anemia, ITP, Graves, good pasture, myasthenia gravis, pernicious anemia, idiopathic thrombocytopenic purpura, and rheumatic fever. I'm assuming these boldened words are the ones that are going to be most focused on. Type 3 hypersensitivity reaction, immune complex, IgG mediated immune complex deposits triggers inflammation, as seen in serum sickness, SLE, which is lupus, RA, post-streptococcal glomerulonephritis, and farmer's lung. Serum sickness. Alright, so that's type 3 immune complex. Type 4, delayed hypersensitivity, T-cell mediated, as seen in Mantoux testing for TB, touching poison ivy, contact dermatitis, chronic transplant rejection. Oh, that's so sad. Type 1 diabetes mellitus, and MS. No antibodies are produced. Okay. Major histocompatibility complex. HLA is part of the MHC with A, B, C genes. I'm going to study this section a little bit more. Do not remember this. Class 1. Antigens are found on all nucleated cells, recognized by CD8 T killer cells. Class 2. Found only on antigen-presenting cells like macrophages and dendritic cells, recognized by CD4. Alright, let's leave it at that since this video has dog barking in it.